Next is Edwin Fong from Novo Nordisk, who will give a talk on the drop-in of concomitant, I always have a hard time pronouncing that, medication. Edwin is recently joined Novo Nordisk and has a background from the University of Oxford Statistics, as well as the Alan Turing Institute. So Edwin, the floor is yours.
Thank you for the intro. So as you can see, my slides are not in Beamer, they're in PowerPoint. And that's going to give a hint to the kind of presentation I'll be giving today. So today I'm going to be giving a more applied talk on an application of longitudinal causal inference, in particular longitudinal targeted maximum likelihood, to address this rather clinical question on how do we tackle the drop-in of concomitant medication. And I'll explain what all these terms mean in the next slide. Just to give a bit of background, this is some joint work I've done with my other colleagues at Novo Nordisk, and also Helene, who just previously gave this talk, was heavily involved in this project and is leading most of the work. So I'm going to start with an introduction for a trial from Novo Nordisk. This is a trial called the SOUL trial. It's a CVOT, which stands for a cardiovascular outcomes trial. So we're going to look at how our drug works in patients with type 2 diabetes. And the outcome in this case will be the time to first MACE. So MACE is a composite endpoint consisting of a few cardiovascular outcomes. So you can think of things like heart attacks, strokes, death due to cardiovascular failure. All of those will be lumped together into a primary endpoint. And we want to see how good is our drug against placebo at preventing patients from developing these kind of cardiovascular complications. So the SOUL trial is still running right now. It'll be ending in 2024. And we're going to be comparing semaglutide against placebo. So semaglutide is our GLP-1, is our main anti-diabetic drug, but also has pretty good cardiovascular risks. And one of the interesting things that comes up a lot in diabetes trials is something called a flexible protocol. So for those who are not familiar, a flexible protocol is a protocol where investigators or the medics during the trial are encouraged to actually prescribe concomitant medication, which is secondary medication, not the same as the main treatment, in order to control things like blood sugar levels and CV risk factors for all the patients, including those in the placebo arm. So that's what I mean by adequate glycemic control. We might see that some patients, for example, in the placebo arm will have that their blood sugar is not in control. The doctors in the trial will be allowed to start other medication that is not semaglutide in our case. So that's what it means. And we call this drop-in of concomitant medication because patients tend to start this during the trial after randomization. And a little bit more context in the medical case, we have this new class of drugs called SGLT2, which is not the same as what we have, our semaglutide drug. And SGLT2 is another type of drug that has a very similar effect. It's glucose lowering, so it improves diabetes, but it also improves the heart health of patients. And because SGLT2 is a concomitant medication, it's not part of the trial, we're going to see some drop-in of this. And as we expect the effect to be quite high relative to our main treatment, this is something that we need to take into account. So I'm going to explain a little bit more in the next slide what this looks like diagrammatically. So in our two trials, we'll have some patients randomized to semaglutide, our drug, and some patients randomized to placebo. Of course, the trial is blind, so nobody knows which group they're in. But in the treatment arm, our drug works pretty well at lowering blood sugar. And so patients will see that, the doctors will see that patients have good glycemic control. And as a result, we'll find that there's no need to start this drop-in treatment called SGLT2 because everyone seems to be very well controlled. However, there'll be some patients, of course, in the placebo arm, and they don't know they're in the placebo arm, but doctors will be checking up on them throughout the trial. And it looks like their blood sugar is very, very poorly controlled. And in this case, due to the flexible protocol of the trial, doctors will be prompted to start a drop-in of some medication. In this case, SGLT2, but perhaps other things too. And we can see that the patients don't know this, but the doctors can measure their A1C, and this will be imbalanced in the trial. And actually, this already has occurred in one of our older trials. So LEADER is an older trial that's similar setup. We're going to be looking at a different drug against placebo called liraglutide. Again, it's a cardiovascular outcomes trial. And here the doctors, this was before the onset of SGLT2, so patients would be using insulin. And we see that between the two arms, so in the solid line, it's the treatment arm, and the dotted line is the placebo arm, a lot more patients in the placebo arm have started using this insulin, this drop-in treatment. And it's a very imbalanced between the two arms. So almost up to 50% of patients are starting by the end of the trial because of precisely this effect we saw at the start. And so long story short, even though we have randomization, it isn't actually enough for balancing this kind of drop-in of concomitant use medication over time. And so we thought this was a really interesting example of where causal inference methods will be needed, even in a randomized clinical trial. So just a bit of intro, I'm sure this is all very familiar to most of you, about a few of the estimates that we've been thinking about. So the usual one that's carried out as the primary analysis for the trial is something called the intention to treat. So here we just randomize, and we're going to treat A0, the randomization variable, as the treatment variable. And then one can imagine that the estimand is treating the treatment arm. We're going to be treating the patients as if they were taking in the treatment arm, and likewise in the placebo arm. And the question that we can imagine answering is that with this prescription of semaglutide, you can expect X% lower risk of cardiovascular events. So note the underline on prescription, which suggests that this is an intention to treat estimand. And then one usually carries out some kind of Cox analysis with the treatment arm as the only covariate, reports a hazard ratio, and then this comparison is protected by randomization. But we're going to go beyond this because we've been working very closely with people who are experts in causal inference. And so some other estimands we can think about, which may depend on who's asking the question. In our case, we have a lot of stakeholders like regulatory authorities, physicians, patients, and other scientists. One of the classic estimands that is often already reported in a lot of the trials is this adherence adjusted effect. So here we're going to be asking the question, if you always take semaglutide as directed, then you can expect this kind of lowering risk. So no drop in treatment for now, just the main treatment. And we're going to be assuming that patients adhere fully. Of course, this isn't the case, but this estimand is usually also targeted in some of the primary trials. And now these next two estimands I'm going to be talking about are going to be much more specific to our drop-in scenario. So one can imagine a regulatory body asking us what is the direct effect of SEMA without any other kind of use of drug. And in this case, this will be without the drop-in treatment SGLT2. So one can ask the question, what is the risk decrease in cardiovascular events if you use our drug, but you don't use SGLT2 at any time? And so that's signified by this kind of always and never for semaglutide and SGLT2 respectively. And one can also imagine a doctor who is interested in what happens if patients take SGLT2 and our drug at the same time. And so if you do take SGLT2, then this is the kind of effect you observe. But the key note here is that the target trials B and C kind of adjust away the imbalance of SGLT2 due to this flexible protocol. I'm going to have a little bit more formality now. Again, still kind of diagramming, but in order to address this kind of question, we actually need to discretize our trial, which is actually a non-trivial problem, but I won't get into that today. But we know that our treatment and drop-in usage and also measurements of blood sugar of each visit is recorded in this longitudinal structure. So we'll have randomization at the start. And then at each visit, one can imagine checking if the patient is taking our treatment, if the patient is taking SGLT2, and then taking a sample of their blood and measuring their blood sugar. So in this case, I've used A to mark the main treatment, Z to mark the drop-in treatment, and L to mark this measurement. And we'll see later on, this is actually a time-varying confounder. And then we carry this out throughout the trial. And then at the end of trial, we can check if the patient has had a MACE event or not. So this is signified by the outcome YK, which is a binary value. And just for some context, L in this case will be a continuous variable. And so to formalize the static intervention that we mentioned before, the question really asking is, what is the probability of MACE, in this case, a potential outcome, I guess, be if the patient took this interventional regime with treatment A bar and drop-in Z bar. So here A bar is a sequence, and likewise with Z bar. And in the no drop-in case, then we'll have this set of A bars and Z bars where we want to look at the treatment arm and intervene so that none of the patients are taking insulin. And similarly in the placebo arm, intervene so that nobody is taking insulin. But what's interesting is that confounding in this case is quite complicated. And this is a very classic example of something called confounded treatment feedback, which we'll talk about in the next slide. And we're going to look at what happens between the visits. So in this simplified DAG here, we're going to be looking at what happens between visit K and K+1 during the trial. So at just before visit K+1, the doctor will be measuring a patient's HbA1c. They'll see how high their level of blood sugar is and decide whether or not to start or stop this drop-in treatment SGLT2 at visit K+1, hence the arrow between the confounder and the drop-in treatment. But we also know from a lot of the trials that we ran before that HbA1c also causes MACE. So those patients who have really high blood sugar, this tends to cause cardiovascular failure, hence the arrow between A1C to MACE. And so this is just a classical confounder DAG where HbA1c is going to be causing both the drop in treatment at visit K+1 and also the outcome at the end of trial. And of course, our treatment also works for the outcome. So this is just a classic confounder DAG. What makes things more complicated in this longitudinal setting is the fact that A1C is also a mediator. So in the previous visit, a doctor would have done something very similar, decided to prescribe the drop-in treatment SGLT2 or not, which would in turn lower or raise HbA1c. And as we just mentioned, this is also going to impact MACE. So we have this arrow going from the drop-in treatment through the confounder to MACE. And so A1C is both a mediator and a confounder. And so classical regression methods will just fail because you want to adjust for A1C to handle confounding, but you shouldn't handle it because you'll be blocking some of the mediation effect. So traditional adjustment fails, and you need to use a little bit more complicated methods. In this case, G computation in the longitudinal setting. And of course, G computation is a pretty old method, but I think it'd be nice to just walk through what this looks like in our setting as well. So a little bit of maths, and this will be pretty much it for the presentation. We can imagine our potential outcomes as being Y superscript S, where S is an intervention both on the main treatment A and the drop-in treatment Z. And so we can pick any sequence we want, but the one that makes most sense for us is for A to be 1 and Z to be 0 throughout the whole trial. And then our treatment effect is going to be the difference between the effect in the treatment arm and the placebo arm. In this longitudinal causal inference, the main assumption is this sequential exchangeability. So we're going to be assuming that the potential outcomes are independent of the treatment at time K, conditioned on history for every time step. And so we need to make sure we're measuring sufficiently enough time-dependent confounders here. In our case, it's kind of a bit easier because the investigators take certain measurements during the trial, and we know that they use those measurements to make the decision to initiate drop-in or not. So we have a relatively safe control for this. And then the next 2 points are pretty classic. It's this consistency and this positivity, which I won't go into too much today, but just a heads up, the positivity is actually a pretty non-trivial problem when working with these longitudinal trials and looking at static treatments. So under these assumptions, our causal estimate is identifiable from the observations through essentially a G formula for longitudinal data. So the expectation of our potential outcome in whichever arm we're interested in is going to be this integral here, where we're going to be integrating over this outcome regression model essentially, and over these confounder distributions. And so this looks a little bit like the typical non-longitudinal G computation. One thing that you can notice is that the confounder distributions would have essentially removed the dependence on one of the treatment nodes, and then we'll be integrating over this high-dimensional confounder distribution. So all of these things can be estimated from the data, and this is the kind of G formula that was introduced by Jamie Robbins back in the day. Now, of course, you can just fit the models for each of these things and then evaluate using Monte Carlo. But in our case, when the confounders are more than one dimension, this is going to be pretty difficult. And so what's done in practice is to use iterated expectations and carry out a sequential regression instead, which I'll introduce in the next slide. Here, we're going to just be fitting a lot of regression models. No need to do any kind of density estimation on the confounders. So I have a little illustration of what G computation with sequential regression looks like, and then we can, of course, step through what the algorithm looks like in practice. So suppose we're interested in the no drop-in estimate in the treatment arm. So here we're going to be setting in our idealized trial all of the semaglutide row to be 1. Everyone's going to stay on semaglutide, our main treatment. And then everyone in the SGLT2 row will be set to 0. So nobody is taking the drop-in treatment. And the first step to actually carrying out this kind of G computation with sequential regression is pretty simple. We're going to regress our outcome, MACE, at the end of trial on everything before it. So we're going to be regressing Y on all the time-varying confounders and all of the treatment histories in both the main and the drop-in treatment, giving us a model QK, which takes us input the entire history of the trial. And then what's neat is that you can do this one-step intervention. So here we're going to intervene at the last visit, but only at the last visit, K-1. We're going to set the treatment to be 1 and the drop-in treatment to be 0. As we mentioned, this is the estimate of interest. And then we're going to pass this through our model and spit out these predictions. So these are going to be predictions that are going to be slightly different to the actual outcomes because A, we're taking an expectation, and secondly, we're intervening on the last visit. And now that we have this vector of predictions for 1 for each patient of size n, we just repeat what we did in the first step. We're going to treat this sequence, this vector of residuals as outcomes at time k-1. Then we're going to do the same thing. So we're going to regress this vector of pseudo outcomes with the history of the trial up until time k-2 now, because we've already intervened out visit k-1. This gives us a model q k-1. And then we repeat this. We intervene at time step k-2, plug in this 1 and the 0, because that's the estimate we're interested in, get this new vector of predictions, and then carry onwards until we reach visit 1. And then at the end, we'll have a model that only takes input the randomization treatment arm, and then we can plug in 1 and we can plug in 0 and take the difference. And then taking the empirical distribution of the empirical average of this model will give us the estimate that we were targeting earlier, which is the expectation of the potential outcome. And so the maths to show this is a little bit hairy, but the intuition is quite nice, where you just intervene one at a time in the visit and continue to carry out this regression. And so what's neat, and this is in connection to Helene's previous talk, is that each regression step can use machine learning, and we don't have to do any fancy density estimation here. And one thing we can do, which I'll briefly discuss a little bit more, is to use this targeted maximum likelihood step. We can essentially tweak some of our regression models to get doubly robust and asymptotically efficient estimates, which has really good properties. So as I discussed before, this is just what the algorithm looks like in print. We have an initial regression step, and then we run this loop where we're going to intervene at each time point. Then we're going to predict a residual of pseudo outcomes from each patient, plugging in the intervention in each time. And then right at the end, we're going to take an average of this sequentially regressed model. In the longitudinal TMLE setting, it's really similar. What we're going to be doing is we're going to be running a regression. But after each regression model, we're going to be doing this TMLE step. So this TMLE step at a high level is going to be setting the empirical mean of the efficient influence function to 0. So we're going to need to also fit propensity score models and then adjust our regression model in a way that the empirical mean of this efficient influence curve is 0. This would give us at the end of our whole longitudinal TMLE setting an estimate that is both doubly robust and asymptotically efficient. And so it uses both the regression models and the propensity score. So if one of them is misspecified, then we have still consistent estimates at the end. One final kind of tricky topic that I didn't delve into too much detail is the fact that in our trials, we tend to have right censoring and also competing risks. So earlier, I just showed YK as being at the end of trial, treating it as a binary outcome. However, it's pretty natural to incorporate competing risks and right censoring in this discretized setting. So we're now going to have a 3 vector for our essentially events. We're going to have YK, which is binary for whether or not the outcome occurred. We're going to have DK, which is binary for whether or not a competing death occurs. So what a competing death is where a patient would die of something that's non-cardiovascular related. So we don't want to attribute that patient to having contributed to the outcome. And then CK is, of course, just censoring. So sometimes patients tend to drop out of trials, or we have administrative censoring at the end of the trial. And one thing to note, of course, is that we're going to be intervening on censoring because we want the estimate to be the case where everyone's observed. But YK and DK have this kind of interesting relationship where knowing one of the values will give deterministic predictions. So in one of the examples here is that if a patient has a MACE at time K, then since MACE is defined to be the first occurrence of this event, sorry, the outcome is defined to be the first occurrence of this event, then of course YK plus 1 will also be 1. So we can code this into our regression models to ensure that this kind of constraint holds. And the opposite holds for competing risks. If a patient has a death that's non-related to the outcome, then of course they can't have the outcome again after they have died. And so DK equals 1 would imply that YK plus 1 is 0 for all the future variables. And so this is something we can take into account when we fit all of our models. So at the end, I'm going to talk a little bit about one of the simulations we did to match the SOUL trial. We're also starting to look at analysis of leader, but unfortunately the results are quite preliminary, so I won't be able to share them today. But to outline the simulation, it's quite similar to the trial setting. I won't go through all of the mathematical equations, but I can give some qualitative descriptions of what the model looks like. So the first thing in our model is we're going to have that this time-varying confounder, HbA1c, of course, is going to be causing the MACE event. So having a high value of L will have a higher chance of causing YK. So you can just think of this as a logistic regression on the YK that depends with a positive coefficient on L. The second point is something that we can tweak. Here we can measure how effective is our drop-in treatment compared to the main treatment at both reducing the time to event confounder and at preventing risk of the outcome. So in this simulation that I'm going to show, we're going to assume that the drop-in treatment is equally as effective. So you can think of this as having the same coefficient in the logistic regression as the main treatment. Of course, depending on what your drop-in treatment is, you're going to want to tweak this value. And the distribution that's probably most of interest for us, and that's why I put this out on the slide, is the probability of initiation of drop-in treatment. So we're going to have this drop-in treatment model depend only on the previous drop-in value and also on the time-varying confounder. So the doctor will see what the value of your blood sugar is and also see if you were in insulin or SGLT2 in the previous visit. And it's just going to be a simple logistic regression, which depends positively on both of those things. So if one were to have really high blood sugar, then taking the x bit of a high number will, of course, give a probability close to 1. Hence, this is supposed to model the investigator being quite likely to initiate drop-in treatment. Similarly, if the patient is already on drop-in treatment, it's going to take some inertia to stop taking the treatment. And so we have this positive coefficient behind the indicator function on whether or not they were on drop-in treatment in the previous visit. And then finally, we have an intercept here, which can govern kind of the overall level of drop-in treatment. And so if we simulate this, we're going to simulate this trial with about 10,000 patients and one-to-one randomization in the 2 arms. We're not going to worry about adherence, but we're going to let the drop-in treatment vary naturally according to this initiation rule. So the first thing to see is, of course, the confounders are going to be very different in the 2 arms. We've enforced so that the drop-in treatment is equally as effective in the main treatment, and our treatment is also going to be effective at lowering A1C. So what we see here is that the confounders start around the same value at the start of the trial, but as we go along the trial, the treatment begins to work and brings this blood sugar down on a log scale. And also in the placebo arm, it goes down slowly, but this is due to the drop-in treatment. But what's more key for us is this plot on the right here, which is going to be a plot of drug use fraction against time. So in the dotted lines, we have the main treatment. As we've mentioned before, there's going to be, we're not going to model adherence. So everyone in the treatment arm is on treatment and everyone in the placebo arm stays on placebo. However, what's interesting is for the drop-in treatment, there's going to be this discrepancy between the 2 arms. Initially, we have about 30% of patients on this drop-in treatment. But as we simulate the trial, the placebo patients, more and more of them start the drop-in treatment. And in the treatment arm, some patients even stop taking the drop-in treatment because their confounder levels have improved so much that the investigators think it's not necessary anymore. So it's quite easy to simulate, and it kind of models what we expect to see in our SOUL trial. And then one can simulate this trial up until visit 5, for example, and then compute the estimates using the algorithms we just discussed before. And one can, of course, also estimate the actual truth by repeating this many times and computing the counterfactual estimands. And so in this figure, we have a plot of 3 estimands and their corresponding estimates. We're going to be looking at the average treatment effect between the 2 arms, where in the first estimand, this all drop-in estimand, we're going to be using the static invention where everyone takes drop-in, so Z bar K is equal to 1. In the second row, we have essentially ZK bar equal to what was observed in the trial. So this is equivalent to not intervening on the drop-in treatment. And you can imagine this as kind of being intention to treat with respect to the drop-in treatment. We only really care about, intention to treat because we're not going to be intervening on the drop-in. And then in the third row, this is the estimand that's most of interest for us. This is going to be the no drop-in estimand, where we force everyone to be on 0. And so a few things to note here. The first is to note that when we carry out primary analysis, it'll be something similar to this ignore drop-in estimand. And we can see that by intervening on this, intervening so that none of the patients start this secondary drug, the estimand can change quite a lot. So the red dot in the third row has moved almost 50% of what the ignore drop-in. So we see an increased effect size because we have now not diluted the effect of this imbalanced drop-in treatment between the 2 arms. And the second thing I wanted to highlight as well is that when we estimate this, we can see that the confidence interval for this no drop-in estimate is much higher than that for the ignore drop-in. And this makes sense because we're intervening. So we're only going to be looking essentially at the patients who have been following our treatment regime. But because we have a flexible protocol, not many patients will be following this. And so I won't talk about this too much, but in one of the big questions we actually still need to address now is this positivity constraint, where we find that it's difficult to estimate the no drop-in estimand without practical positivity violations. And we're currently applying this to LEADER. So this is the cardiovascular trial I mentioned at the start. And then when the SOUL results come out, we'll be applying this to SOUL as well. So to summarize my talk, we found that there are actually many other secondary estimands which are helpful for handling this drop-in imbalance and require longitudinal analysis. We're now going to start investigating more complicated dynamic or stochastic treatment regimes where we can kind of answer the similar question, but with better positivity, hopefully. A second point I wanted to make, which was more general, is that even in randomized control trials, we found that causal inference methods are highly needed because there's still many questions that you have to use causal inference methods to answer, since in practice, data is really messy, even with randomization. And finally, a point we've noted as well is that when trying to apply these methods, a lot other practical problems come up. So things like positivity, as I just mentioned before, also numerical stability and model misspecification are things that we now have to think about. Of course, these were things we should have thought about at the start, but by working with this causal inference framework, we've been able to kind of see these more transparently. And so that's it from me. Thank you so much for your time and for inviting me to give this presentation. And I'm happy to take any questions now.
Thank you, Edwin. Questions? I would also just like to remind everybody, I have one, which I will need to, but I would like to remind everybody that there's a Q&A function. So also during the trial, during the talk, you can write a question there, and then I will take it up at the end. But I just had a question about typically when we do these kind of trials, we are somewhat focused on studies that we don't invent the hypothesis when we see the data. But on the other hand, there must be something about which questions, which scenarios we can compare must relate to the kind of behavior that was actually seen, which is probably unknown at the onset. Is it problematic, or have you thought about sort of inventing the scenarios when you see what is available, the variation, when you see the positivity constraints, essentially?
Yeah, that's a really good question, and something we've been thinking about. One thing that we've been advised from sort of the experts in this field is that one can do outcome blind simulations. So once you receive the trial, you don't look at any of the event rates, none of the Ys essentially. You only look at the As and maybe some of the time varying confounders, the Ls. And in that case, you're not biased towards an effect that you want to see, but you can still address all of the things like positivity. And you can even sort of fit models that you think maybe using historical data to simulate some of these outcomes, and then you can see if the estimates actually change a lot. And one final point to that is that in regards to positivity, we're kind of hoping to have a few estimates, not just the no drop in one or the intention to treat, maybe something in between, where we can also assess whether or not this estimate is reliable based on some estimate of positivity violation. Again, tips from those great folks at Berkeley.
Okay, yeah, thank you. Other questions? If not, please join me in thanking Edwin.